Now it's my pleasure to introduce the last speaker of today, Dr. Jackson Hoffman. So Jackson did his undergrad at the University of Illinois Urbana before moving to the University of Illinois Chicago for his PhD. There he conducted his thesis work in the lab of Bradley Merrill, where he worked in ES cells and mouse models to demonstrate a critical role for TCF7L1 in regulating the transition between pluripotent states and differentiation. Jackson then moved to North Carolina for his postdoc, joined the lab of Trevor Archer at the NIH, the National Institute of Environmental Health Sciences. There he has conducted several incisive studies on the mechanisms of GR and BRG1 function and hormone-dependent transcription. In 2018, Jackson transitioned from a postdoc to a biologist position, taking on greater responsibility in the Archer lab. Looking at Jackson's CV, his numerous publications, of course, jump out, but I'll just quickly highlight his sustained efforts on committees and working groups, including his work on the Pride at NIHS Working Group and the Special Act Award he received recognizing his contributions to the Diversity, Equity, Inclusion, and Accessibility Working Group. With that, I'll just finish by saying that Jackson is actively recruiting postdocs for the Archer Lab, so definitely reach out to him if you're interested. And now, please join me in giving a warm, albeit virtual, welcome to our final speaker of the day, Dr. Jackson Hoffman. Jackson, it's over to you.
Ben, thanks so much for the wonderful introduction. You make me sound better than I am. Let's make sure I'm sharing the right thing here. Okay, so yeah, it's just really a pleasure to be invited to speak here, and I've had the wonderful experience of meeting several of our graduate nucleosome organizers at a conference just a couple months ago, and so I'm really excited to be here to talk to you today about a project that is a bit different than the GR and BRG1 and breast cancer project that I've been working on for the last 5 or 6 years. And this is a project that my colleague in the lab, Ginger Muse, and I have initiated and are in part continuing from a previous postdoc at the lab to try to understand how chromatin accessibility regulates the ability of human embryonic stem cells to specify different lineages. And so I'll start briefly by showing you this beautiful picture of the BAF complex. And so the BAF complex is this large multi-protein machine that squeezes onto nucleosomes and utilizes the hydrolysis of ATP by BRG1 to remodel nucleosomes and evict the histone proteins from the DNA. And in doing so, it can open up the chromatin to allow the transcriptional machinery or transcription factors or other things to bind. And so we're interested in BRG1 and its corresponding subunits in the context of human embryonic stem cells because BRG1 and BAF subunits are mutated in a variety of neurodevelopmental diseases, including autism spectrum disorders and Coffin-Siris syndrome. And BRG1 has been shown to have context-dependent tumor suppressive and oncogenic functions in a variety of really severe and aggressively malignant neurodevelopmental tumors. Highlighting here the atypical teratoid rhabdoid tumors, which uniformly show a biallelic loss of either BRG1 or BAF47. And so understanding how these mutations in the BAF subunits can give rise to these tumors is really critical for human health. And so before moving on too much further, I want to introduce the types of progenitor cells that give rise to these tumors. And so largely, these tumors are thought to arise from neural progenitor cells or neural stem cells during embryonic or postnatal development. And so in the Archer lab, we utilize a number of different cell types. We start with human embryonic stem cells, which are derived from the early human embryo, where the cells that will contribute to the adult body consist of a single layer of cells that's fully pluripotent. And after a few additional days of development, this layer of pluripotent cells undergoes gastrulation and the formation of the 3 primary germ layers. And over here at the anterior side of the embryo, the ectoderm eventually starts to undergo morphological changes as it's proliferating, and it forms this structure called the neural tube. And you can think of the neural tube sort of as the residence of the neural progenitor cells in the developing embryo. And at the dorsal end of the neural tube, there's this really other interesting form of neural progenitor cell called neural crest cells. And these are really intriguing because they're very highly migratory, more mesenchymal type of stem cell that is derived from the neuroectoderm lineage. And so we're really interested in this because the mutations that occur in BRG1 or its other subunit BAF47 are not only involved in neurodevelopmental tumors, but in a whole host of different syndromes and diseases in human health. And a lot of these, including rhabdoid tumor predisposition syndrome, are really aggressive and really malignant, and are primarily found in toddlers and in infants. And interestingly, the mutations in these two subunits are mutations in the proteins of the BAF complex that actually form contacts with the nucleosome. And so that, argues that the chromatin remodeling functions of the BRG1 BAF complex are most likely required during the specification of the neural progenitor cell types that would eventually give rise to these types of tumors. And so with that sort of hypothesis in mind, Lee Langer, a previous postdoc in the lab, took it upon himself to look at what happens when you deplete SMARCB1, or BAF47, as it's also called, from human embryonic stem cells. And when he did that, he found that depletion of BAF47 in human ES cells or during differentiation results in about 20,000 regions of the genome that have differential chromatin accessibility. About 2/3 of those lose accessibility and about 1/3 gains accessibility. And this was concomitant with what appeared to be a failure in neuronal or ectodermal differentiation. And when he associated these differentiation pathways with the differential accessibility, he saw that it looked like BAF47 depletion was locking the cells into a pluripotent state, where these cells had a failure to lose accessibility at human embryonic stem cell specific superenhancers. And they also had a failure to gain accessibility at enhancers that were specific or had accessibility specific in neural tissues. However, this doesn't necessarily paint the entire picture, because a few years later, I did some IP mass spec for BRG1 in human ES cells, and I found that knockdown of BAF47 doesn't actually have much of an effect on the total overall levels of BAF complexes that we can detect in human ES cells. It does have some bit of effect on the flavor of BAF complexes that we find in these cells, where we have a slight decrease in the predominant BAF complex and slight increases in the PBAF and NC-BAF complex, which you can also see in the fold change of the percentage of complexes that contain the PBAF and NC-BAF specific subunits. But in general, we've still got BAF complexes around when BAF47 is depleted. And so that's what led Ginger and I to start investigating what the role of BRG1 was during this process. And so the model system that we use is we start with human embryonic stem cells, and we subject them to 9 days of dual SMAD inhibition to inhibit both TGF-beta and BMP signaling. And over the course of this assay, you get a pretty uniform population of neural progenitor cells. And so for the purposes of our experiments, we manipulate BRG1 activity and expression in a number of different ways. I'll primarily show you BRG1 shRNA, where we can pre-treat for 3 days to deplete BRG1 at the onset of this differentiation experiment, and then maintain depletion throughout. We've also recapitulated all of these experiments with PROTAC, which more rapidly depletes BRG1, and we largely see the same effects in both experiments. And so one of the first things we did was look at these earlier neural progenitor cells at day 3 and perform a TAC-seq to determine what regions of the chromatin were losing or gaining accessibility when we deplete BRG1 in these cells. And so when we take all of our TAC-seq data, which we performed with biological triplicate in these 3 samples, we can then call peaks from nucleosome-free regions where we have fragments that are 100 BP or less. And what you can immediately see is that there's a significant loss of nucleosome-free regions when BRG1 is depleted, either by shRNA or by PROTAC. And if we limit these down to the high confidence peaks that are present in multiple replicates, we can see that while approximately 90,000 peaks are common to all of the treatments, we have about 40,000 nucleosome-free regions that are specific to the control cell types, or the control cells. And so those represent sites that are dependent on BRG1 for chromatin accessibility. If we take these 164,000 sites and we do feature counts and look to see what's differentially accessible, we can see that there's a huge shift, like 83% of the first principal component is represented by the depletion of BRG1, either by PROTAC or shRNA. And we look at what sites are significantly decreased or increased. We can see that in both the BRG1 knockdown and the PROTAC, we lose 30,000 plus sites of accessible chromatin when we deplete BRG1. We also gain a smaller number, about 2,000 or 3,000 sites of accessible chromatin. And importantly, you can see when we compare the nucleosome-free regions detected in the two different versions of knocking out or knocking down BRG1, you can see that they're largely very similar. And the only things that really pop out as significant are things that are a little bit more decreased when we use the PROTAC. And so what are these regions? If we simply do some annotation of these based on where they're located in relation to gene TSSs, from all peaks and the non-change peaks, you typically get about a third of the nucleosome-free regions around gene TSSs, and then about 2/3 in intergenic or intronic regions, suggesting that they're enhancers. And the sites that are significantly different in the nucleosome-free regions that are lost in the BRG1 knockdown or the PROTAC are much more enriched for sites that would most likely be enhancers. And if we do motif analysis under these, the top 6 motifs are just ridiculously enriched for the SOX motif. And so this is really interesting because SOX2, for instance, is a pioneer factor in embryonic stem cells and in neural progenitor cells. And a number of the SOX factors are critically involved in patterning the differentiation and specification of neural lineages. And so we know that there are significant changes to the chromatin accessibility landscape at the earlier time points of this neural progenitor cell differentiation pathway. And so we wanted to continue doing experiments to look to see what's happening to these cells as they progress through the pathway. And so the first and easiest thing we did is simply look at cell morphology at day 6. In our control cells, we get this uniform lawn of neural progenitor cells. However, in our BRG1 knockdown cells or in our PROTAC treated cells, we start to find these regions of the lawn of cells where it's disrupted. And instead, you get these much flatter and mesenchymal looking cells forming in BRG1 knockdowns. So as these neural progenitor cells form, by about day 4, 5, or 6, these cells should be uniformly SOX2 positive, and they should all have gained the expression of PAX6, which is another critical transcription factor in neural progenitor cells. And as we progress through this pathway, by day 9, you should have pretty much 100% neural progenitor cells, with the exception of about 5% of the cells that spontaneously progress towards neurons or neuronal lineages. And so Ginger took these cells. She took our human embryonic stem cells and our NPCs at day 6 and performed some flow cytometry for SOX2 and PAX6 to determine whether we were forming the correct population of neural progenitor cells. And so in these flow plots, the wild type cells are outlined in gray, the BRG1 depleted cells are outlined in blue. And you can see human embryonic stem cells are fairly uniformly SOX2 positive and PAX6 negative. By day 6 of the neural progenitor cell differentiation process, the wild type cells maintain SOX2 expression and they uniformly upregulate PAX6. But we were surprised to see that the BRG1 depleted neural progenitor cells have populations that are either SOX2 negative or PAX6 negative, which normally we don't see at all in the wild type cells. And if we look at these two together, you can again see that the control cells are almost uniformly double positive for SOX2 and PAX6, whereas the BRG1 depleted cells have a double negative population, as well as a PAX6 negative SOX2 positive population. And these are populations of cells that we would not expect to arise in this experiment. And so to try to track down what these populations of cells are that are forming in our assay, we decided to perform single-cell RNA-seq at each of our 3 time points of neural progenitor cell differentiation. When we do this, we were really surprised to see that the BRG1 knockdown cells are completely distinct from the control cells. We may have expected some of the cells to cluster or to overlay the wild type cells and then just these different populations to be distinct. But we really see that all of the cells in which BRG1 has been depleted is distinct. We also observe in the wild type cells, the day 3 and 6 and 9 neural progenitor cells cluster distinctly from one another. However, in the BRG1 depleted cells, the day 6 and the day 9 cells form intermixed clusters, suggesting that we're really getting something different happening in these cells. And so to try to investigate this further, we called de novo clusters and used these to try to identify the different cell types that might be arising. And one of the first things we looked at is just whether or not we're getting this spontaneous neuronal differentiation in these experiments. And so like I said, in wild type cells, this cluster 10 represents these spontaneously forming neurons. And we see about 5% of the cells, as you would expect based on previous literature. However, in the BRG1 knockdown cells, this cluster of spontaneous neuronal differentiation is much larger, and we're getting up to 10 or 11% at day 6 and day 9. And you can see these also with markers of early neuronal lineages. However, the BRG1 knockdown cells seem to be forming a different kind of neuronal lineage, in that they lack subnormal markers like FESF1 and PRNT8, but gain markers of other neuronal lineages such as ASTM2 and SCNG. And so beyond these differential neuron populations, it seems that BRG1 knockdown is also inducing a different type of neural progenitor cell. And so this nicely recapitulates our FACS data, where we see these differential populations of PAX6 negative and PAX6 SOX2 double negative neural progenitor cells. And if we look at some other markers of neural progenitor cell specification, such as PAU3F1, we also see that there's a depletion or a loss of expression of these factors in the BRG1 knockdown cells. We've also looked at markers of neural progenitor cell maintenance, or cells or markers that are more involved in the self-renewal of neural progenitor cells. And we also see with some of these, we have a complete loss of the expression of these factors in the BRG1 knockdown cells. We can also look at some cell surface markers that are indicative of different lineages. And so typically our wild type cells express CDH8, which is seen as a marker of neurogenic progenitors, whereas our knockdown cells express CDH6, which is seen as a marker of gliogenic progenitors. And so it seems as if we're really getting a different type of NPC. And that is further underlined by the fact that we see different patterns of transcription factor expression in the BRG1 knockdown cells. Here you can see 3 factors, ZIC1, PAX3, and MSX1, which we largely don't detect in the wild type cells, but are robustly detected in the day 6 and day 9 BRG1 depleted neural progenitor cells. And this is really interesting because these transcription factors all have a pattern or are all expressed in the dorsal neural tube during development. And here's just an example from a paper looking at the expression of PAX3 and PAX6 in the mouse neural tube. And so PAX6, which is our more normal neural progenitor cell marker, it's expressed along the middle of the mouse neural tube. But as you move to the dorsal side of the neural tube, you lose PAX6 expression and gain PAX3 expression, which looks strikingly similar to our neural progenitor knockdown cells. This is also really interesting because this dorsal side of the neural tube is where the neural crest is specified, and cells at this dorsal tip of the neural tube will delaminate, undergo an EMT, and form this special population of stem cells called the neural crest. And so we looked to see whether we could detect markers of the neural crest in our BRG1 knockdown cells. And indeed, in this cluster 5, we have specific enrichment and expression of a variety of master regulators of the neural crest lineage, such as FOXP3 and ETS1. And so this really fascinated me. I've been, as Ben mentioned, I started out doing some embryology in grad school, and I've always been fascinated by the neural crest because it's a really cool cell population. It's highly proliferative. And it gives rise to a wide variety of cell types throughout the entire body, including our cartilage, most of our peripheral nervous system, melanocytes, and Schwann cells. And so pretty much all of the cells that are derivative of the neural crest are SOX10 positive. And so I now wanted to look at these cluster 5 cells to see if I could detect any signs of them precociously differentiating along these neural crest lineages. Oops, sorry. And so if we look at this cluster 5, we can see that it specifically is enriched for cells that have SOX10 expression, which is this marker of neural crest derivative cell types. And if we look even further along the pathway, we can begin to see markers such as MIT-F, which is a gene exclusive to melanocytes, and NPZ, which is a Schwann cell marker gene. And the expression of these genes are really only detected in these SOX10 positive cells in this cluster 5. And so it certainly seems that we're seeing precocious differentiation of these cells down neural crest lineages. And so to sum this up, in normal human embryonic stem cells, inhibition of BMP and TGF signaling gives rise to a uniform population of neural progenitor cells, and you see some spontaneous differentiation to neurons. However, when we deplete BRG1, we get huge shifts in chromatin accessibility, with about 30,000 nucleosome-free regions being lost or decreased. And this gives rise to a different flavor of neural progenitor cells with heterogeneous populations of PAX2 and PAX6 and SOX2 expression, as well as expression of other factors like ZIC1 and PAX3. This in turn leads to an enhanced progression to neuronal lineages, as well as different neuronal marker gene expression, and also precocious formation of neural crest cells, and even potentially the formation of cells of other neural crest lineages. And so as we follow up with these experiments, we're really interested in trying to understand what this limited chromatin accessibility that we observe at the early time points of neural progenitor cell specification is actually doing to cause this change in the neural progenitor differentiation pathway. And we also really want to try to relate this to the role that BRG1 is playing in neurodevelopmental diseases. And we're hoping to do that by extending this into other model systems in the future. And so with that, I'd like to thank my boss, Trevor Archer, for giving me the leeway to work on this project, as well as several other projects in the lab. Ginger Muse for being my collaborator and co-author in this project. Isabella Gondara, who is a brilliant summer student that worked for us for several weeks and helped analyze some of the single cell RNA-seq data. Lee Langer for initiating this project with BAT47 way back in the day. And then the rest of the Archer lab, as well as the other labs at NIEHS. And as we said at the beginning, our lab is currently looking for postdocs, as are several other labs in our department that all have really interesting and amazing projects. And you can email me or send me a message on social media, and I'd be happy to answer any questions. Thanks so much.
Awesome. Thank you, Jackson. That was fantastic. I'll start off with a question. I was wondering, so with your BRG1 depletion, you get kind of like an altered NPC state. And from there, it goes on and you get kind of neural crest and some other kind of weird neuron plus lineages. Have you thought about restoring BRG1 expression after you get to the altered NPC? And like, is the subsequent things you're seeing as like the altered state there, or is it the continued need for BRG1?
Yeah, that's great. Yes, we definitely have thought about that. We're currently performing several different like pulse experiments to look to see at what point in this process is BRG1 required. And preliminarily, it looks as if BRG1 is needed right at the initial time point, and that we can relieve the depletion and we still get these same effects.
Interesting. And when in this time course does BRM expression start to go up?
So we've done some Western blots across the time course, and we have obviously RNA data, and the levels of BRM never really come up to more than like 1/10 of what it looks BRG1 is at. And so I don't think there's too much BRM in the system, but yeah.
Yeah, and that would make sense with why your knockdown of BRG1 looks so similar to the PROTAC.
Yeah, exactly.
So a number of questions. First one says, very nice talk. Have you looked at how BRG1 knockdown affects genes important for EMT?
Yeah, we have a bit. And so several of the genes involved in EMT, including most of the transcription factors that drive EMT, are also enriched in the population of cells that has that looks like neural crest cells. And so we think that there probably is some involvement there, but we don't know yet whether that's a direct effect of BRG1 or if it's just a sign that it's predisposing the cells to this type of neural crest lineage.
And then we've got a question about the single cell RNA-seq data asking if you tried integrating the data. I'm not a single cell person, so I assume that's analysis approach.
Yeah, so I have. I've played around with the data a lot. And I've tried even like regressing out the differences between the cell lines. And if you really manipulate the data, you can start to get some of the wild type cells to overlap with the BRG1 knockdown cells, but it feels like way too heavy-handed of an approach at dealing with the data. It seems like the cells really are just that different.
Great. Which, I mean, it makes some sense to me because it's such an integral, like important chromatin regulator that
right.
I'd expect it to be pretty profound effects. So Emily Brown says she may have missed this, but wonders why you chose days 3, 6, and 9 as your time point harvest along the differentiation.
Yeah, that's a good question. So those are just kind of the typical time points that people look at when they're tracking neural progenitor differentiation. We have looked at additional time points, and largely day 3, 6, and 9 seem to be representative of what we see throughout the time course.
And then a final question is, how can we control the population of non-neural cells during culturing the NPSC or NSCCs?
Yeah, so that was the first thing we thought, was that since we were getting SOX2 negative or PAX6 negative populations of cells, we immediately thought, oh, the cells are just dying, or oh, they're becoming mesoderm and endoderm. And really, we don't see any evidence of that. So it seems like these are all progressing down neural lineages. It's just they're becoming different neural lineages.
Yeah, that's one of something that I found just absolutely fascinating, seeing all these different types of neuronal cells appearing.
Mm.
Okay, so that's all the questions, and we're actually right on time, which is fantastic. So thank you, Jackson. Thank you to our other 3 speakers, and thank you everyone for attending. This was a kind of a fun alternative format. I got a lot of it. I hope everyone else did too. And so just thank you. And we'll be back with a normal seminar in 2 weeks, which will feature Ulditz, one of our co-organizers, sharing her work.
Thank you all very much.
Thank you.